Right now, before you eat anything, take a glass of plain water and imagine it moving down through the throat into an empty stomach. No sugar, no salt, no caffeine, just a low viscosity fluid entering a system that has been running for hours without intake. Your brain detects that event before the water has fully entered your blood. That is the first thing to understand. The water does not instantly become brain fuel. It does not wash toxins out of the skull. It does not flip a magic switch behind your eyes. The mechanism is colder than that. Cleaner, more mechanical. You have added volume to a hydraulic network. You have changed the signal coming from the mouth, throat, stomach, gut, blood, kidneys, hypothalamus, and autonomic nervous system. You have not given the brain a miracle. You have given it information, and the brain is a control system built to act on information before the chemistry has finished catching up. After a night without drinking, your body is usually not in emergency dehydration. In a healthy adult, it is more often a small drift. Water has left through breathing, skin, urine, and sweat. The blood is still blood. The brain is still protected, but the fluid ratio inside the system has changed slightly. The solvent has fallen. The solute concentration has risen. That ratio is called osmolality. A typical regulated range is roughly 280 to 295 milliosmoles per kilogram, and the brain has specialized osmoreceptor circuits that help control thirst and arginine vasopressin release when the concentration shifts. Think of it as voltage in a fluid circuit. Too much dissolved material per unit of water, and the voltage rises. The sensor does not need your opinion. It measures the gradient. The hypothalamus and nearby circumventricular organs read the condition of the blood like a pressure gauge, reading a pipe. Two regions matter here, the subfornical organ and the organum vasculosum of the lamina terminalis. They sit near the third ventricle, and unlike most brain tissue, they are positioned to sample blood chemistry more directly. They are not mystical thirst centers. They are exposed instruments. They measure the fluid. When you drink water on an empty stomach, the first change is not hydration. It is prediction. The mouth, throat, and upper gut send a fast signal that water is entering the machine. Thirst can begin to fall within seconds, long before the water has had enough time to correct blood volume or plasma osmolality. That means the brain is not waiting for the final blood test, it is forecasting. A thermostat does not wait until the entire house reaches the target temperature before turning the heater down. It reads the slope. It sees the direction. Your thirst circuitry does something similar. As water crosses the tongue and throat, sensory input begins suppressing the drive to drink. Then the stomach and intestine provide the next layer of confirmation. The system anticipates correction before correction is complete. On an empty stomach, that signal is cleaner. Food changes, gastric emptying, fat, fiber, protein, and osmotic load slow movement. Plain water in a fasted stomach is handled differently. The stomach is not grinding a dense meal. It is passing a simple fluid load downstream. A fasted state MRI study of a 240 milliliter water dose reported a gastric half-emptying time of about 13 minutes, with stomach volume returning toward baseline by about 45 minutes. That is not instant, but it is fast enough for the brain to treat water as an incoming correction. The stomach stretches, mechanoreceptors detect wall tension. The vagus nerve carries traffic from the gut toward the brainstem. The brainstem relays that information into networks that regulate arousal, autonomic tone, and internal state. This is not thinking. This is instrumentation. A wall stretches, a nerve fires, a nucleus receives, a control loop adjusts. The empty stomach makes this easier to understand, because the input is simple. A known fluid enters an empty chamber. Volume rises. Pressure distribution changes. Transit begins. Sensors report movement through the pipe. But the brain is not only tracking the stomach, it is tracking water demand across the whole body. During the night, vasopressin may rise to help conserve water. Vasopressin is the antidiuretic hormone, also called arginine vasopressin, and its main job in this context is to tell the kidneys to save water. It does this by acting on V2 receptors in the kidney collecting duct, increasing cyclic AMP signaling and promoting aquaporin 2 water channel insertion into the cell membrane. That is the valve mechanism. More aquaporin 2 in the membrane means more water can move back into the body instead of being lost into urine. The kidney is not a passive filter. It is a variable valve bank controlled by endocrine voltage. When you drink water, the system begins moving toward lower vasopressin demand, not because the water has performed a cleansing ritual, but because the control loop now expects incoming solvent. The blood concentration can be corrected. The kidneys can eventually reduce conservation. Urine can become less concentrated. The command changes because the inputs change. This matters to the brain because the brain is expensive tissue. It is only about 2% of body mass, but it consumes about 20% of resting energy. It depends on blood flow, glucose delivery, oxygen delivery, ion gradients, and the precise electrical behavior of membranes. Neurons do not fire in a vacuum. They fire inside fluid. Every spike is electrochemistry. Sodium moves, potassium moves, calcium moves, chloride shifts, pumps restore gradients, astrocytes buffer the environment. Blood vessels adjust diameter. Cerebrospinal fluid circulates. The whole system is a wet electrical machine. When hydration drifts, cognition does not usually collapse. It becomes more expensive. That distinction matters. A mildly dehydrated brain may still complete the task, but it may require more activation, more effort, and more compensation. In MRI research, dehydration has been associated with changes in brain tissue volume and task-related activation patterns, suggesting the system may work harder under fluid stress. The output can look normal. The energy cost may not be normal. This is why drinking water after an overnight fast can change performance in some people and do nothing obvious in others. The effect depends on the starting condition. If you wake already well hydrated, with low thirst and stable osmolality, adding water is a small input. If you wake thirsty, concentrated, sleep-restricted, or under heat stress, the same glass becomes a larger correction. The machine state matters. Caroline Edmonds and colleagues tested adults after they had fasted and avoided fluid intake since the previous evening. In one condition, participants drank 500 milliliters of water before testing. In another, they did not. Reaction time improved after water, and subjective thirst moderated the effect. The thirsty participants were the ones whose speed changed most. That is the honest result. Water did not turn every brain into a sharper brain. It removed a constraint in the brains that were carrying the thirst load. In media summaries of that work, reaction times were described as up to 14% shorter after drinking water, especially among participants who felt thirsty. But the same research also reported that one complex rule-learning task became slightly worse after drinking. That detail is important. The body is not a motivational poster. It is a system with trade-offs. A simple reaction time task is close to a sensory motor circuit. Detect the signal, press the button. Lower background thirst load may reduce interference, but complex rule learning is a different machine. It requires working memory, inhibition, updating, error correction, and sustained control. A sudden internal state change may not help every circuit in the same direction. One glass of water is not a universal cognitive enhancer. It is an input into a control system. But there is a second mechanism operating at the same time. When water enters the gut, it can trigger an autonomic response. This is not about intelligence. It is about vascular control. Blood pressure, vessel tone, sympathetic outflow, and baroflex behavior are all part of the same mechanical environment that supplies the brain. In 2000, Jens Jordan and colleagues published work showing that water drinking can produce a strong pressor response in people with autonomic failure and a smaller response in some older control subjects. In healthy young adults, the effect is usually much smaller. Later analyses suggest a rapid 350 to 500 milliliter water bolus may raise systolic blood pressure only slightly in healthy participants by about 2.33 millimeters of mercury on average, and diastolic pressure by about 2.73 millimeters of mercury. Small numbers. But small numbers are not meaningless in a regulated hydraulic circuit. The brain is protected by autoregulation. Cerebral vessels constrict and dilate to keep blood flow stable across ordinary pressure changes. But the system still cares about pressure, volume, posture, and sympathetic tone. When you stand up in the morning, gravity moves blood downward. The heart must pump against a vertical column. Baroreceptors in the carotid sinus and aortic arch detect stretch. Sympathetic signals tighten vessels. Heart rate may rise. Venous return must be maintained. You experience this as alertness or lightheadedness. The machine experiences it as flow management. A glass of water on an empty stomach adds an osmotic and volume signal that can support this vascular control loop, especially in people prone to orthostatic lightheadedness. This is why water bolus ingestion has been studied clinically in autonomic failure and orthostatic hypotension. But in a healthy person, the effect is not usually dramatic. Again, calibration matters. Not cure, not superpower. Control loop. Now add the third layer. Thirst itself consumes attention. Thirst is not just a message in the mouth. It is a motivational state. It pulls computation. It makes the brain monitor the body, search for water, evaluate discomfort, and prioritize correction. That means thirst can compete with other cognitive tasks. This is the bottleneck idea. If one control system is constantly sending an error signal, other systems must process around it. You can still work, you can still read, you can still make decisions, but part of the brain is carrying an unresolved internal alarm. Drinking water can reduce that alarm before the blood is fully corrected. The subjective feeling of thirst may fall quickly, the motivational load may drop, the task circuit may now run with less interference. This is why the water effect often appears strongest in people who begin thirsty. Not because water has added intelligence, because it has removed noise. A circuit with less noise does not become magical. It becomes easier to read. There is a fourth layer, and it sits deeper in the tissue. Brain cells live in an extracellular fluid environment where ion concentration, osmotic pressure, and cell volume matter. If extracellular fluid becomes more concentrated, water tends to move out of cells. If it becomes less concentrated, water tends to move in. The brain has protective mechanisms against rapid swelling and shrinking, but the principle remains physical. Water follows osmotic gradients. A neuron is not a wire in dry air. It is a membrane-bound electrochemical device surrounded by salt water. Its firing threshold depends on gradients. Its recovery depends on ATP-powered pumps. Its network timing depends on chemical conditions. A small systemic shift does not automatically disrupt firing, but the system is built around narrow tolerances. That is why osmoregulation exists. The body spends energy to protect the water ratio, because electrical tissue is sensitive to fluid physics. The brain is not floating above the body, it is submerged inside the body's chemistry. When you drink water on an empty stomach, the correction pathway begins at the top. Mouth, throat, stomach, intestine, blood, hypothalamus, pituitary, kidney, vessel wall, brainstem, cortex. Each layer has its own clock. The mouth reports in seconds, the stomach reports in minutes, the intestine absorbs over minutes, the blood chemistry shifts more slowly, the kidney output changes over tens of minutes to hours. The subjective feeling may change before the laboratory value changes. That timing difference is the whole story. The brain is not reacting only to what is true now, it is reacting to what the body has reason to believe will be true soon. This is predictive control. Engineers use it when a machine adjusts before a measured variable reaches its final value. Biology uses it because waiting can be dangerous. If thirst were only shut off after the blood became perfectly diluted, you would overdrink. If it never anticipated absorption, you would keep drinking long after enough water had entered the pipeline. So the brain estimates. It uses the mouth to estimate intake. It uses the gut to estimate fluid type. It uses the blood to measure actual concentration. It uses the kidney to correct output. The most precise part is not the glass. The most precise part is the matching. Drink pure water, and gut osmosensors can help confirm that the incoming fluid will dilute. Drink seawater, and the signal is different. The volume is there, but the osmotic correction is wrong. A control system that only counted swallowing would fail. The brain does not only ask, did fluid enter? It asks, what will this fluid do to the internal ratio? Research on thirst circuitry has shown that gut-to-brain signals about fluid osmolarity help sustain the rapid inhibition of thirst neurons after drinking. This is elegant because it is mechanical. The system reads the properties of the input. It does not believe the input, it tests it. Now think about the phrase, empty stomach. The phrase has been turned into folklore. People say water on an empty stomach detoxes the body, melts fat, activates organs, cleans the colon, wakes the brain, resets the metabolism. Most of those claims are overbuilt. The evidence does not support the mythology. But the empty stomach still matters mechanically. It means the water is not competing with a dense meal for gastric processing. It means the stretch signal is easier to isolate. It means absorption may begin quickly. It means thirst and hydration state, from overnight abstinence, may be more relevant. It means a cognitive test performed before breakfast can reveal the interaction between thirst, fluid intake, and attention. The accurate claim is narrower. If you are thirsty or mildly underhydrated after the night, drinking plain water before food can reduce thirst signals, begin correcting osmolality, influence autonomic tone, support vascular stability, and sometimes improve speed or decision-related performance. If you are already hydrated, the brain may not change in a way you can feel. That is not disappointing, that is precision. The body does not reward unnecessary correction, it corrects error. This is why drinking too much water is not better. The system has limits. Excessive water intake can dilute blood sodium, a condition called hyponatremia, and severe cases can be dangerous because water shifts into cells, including brain cells. The same physics that makes water necessary can make too much water harmful. Dose matters, context matters, kidney function matters, medications matter. Heart failure, kidney disease, endocrine disorders, and some psychiatric conditions can change the safe range of fluid intake. A healthy person drinking a normal glass of water in the morning is one scenario. Forcing liters of water, because a video said the brain loves it, is another. The machine has specifications. Respect the specifications. Now return to the brain itself. When you wake, the brain is not simply turning on, it is changing state. Sleep inertia may still be present. Adenosine pressure may still be clearing. Cortisol is rising as part of the circadian awakening response. Blood pressure is shifting upward. The autonomic system is preparing posture. The gut is quiet but not offline. The kidneys have been concentrating urine. The mouth may be dry. The thirst circuit may be signaling. A glass of water enters that transition, not as a stimulant, as a stabilizing input. Caffeine pushes receptors. Sugar changes glucose and insulin dynamics. Salt changes osmotic load. Water is simpler, it changes volume and dilution. That simplicity is why its effects can be subtle but fundamental. It is not a drug. It is the solvent the drug would dissolve in. And because the brain is a high-frequency electrical organ, solvent status matters. Reaction time, mood, fatigue, attention, and memory have all been investigated in hydration research. But the literature is mixed. Some studies show benefits after water intake. Some show effects only in thirsty people. Some tasks do not improve. Some complex tasks can move in the wrong direction. This is what honest biology looks like. A real system does not produce one clean marketing sentence. It produces conditional effects. The effect of drinking water on the brain depends on baseline hydration, thirst, temperature, sleep, age, blood pressure regulation, kidney response, stomach contents, and task type. That is why the strongest statement is also the least viral one. Water helps most when lack of water is part of the bottleneck. But when it is part of the bottleneck, the correction can feel immediate because the brain lowers the alarm before the chemistry is completely restored. You feel clearer. The underlying event is not clarity, it is reduced error signaling. You feel more awake. The underlying event is not energy creation, it is autonomic adjustment, sensory prediction, and lower cognitive interference. You feel steadier. The underlying event is hydraulic support. These are not poetic differences, they are engineering differences. The heart is a pump. The vessels are variable diameter tubes. The kidneys are filters with adjustable water channels. The hypothalamus is a regulatory controller. The mouth and gut are input sensors. The brain is an electrical load sitting inside a fluid-cooled housing. Water is not a symbol in that machine, it is the working medium. This is why the same simple act can be ordinary and significant at the same time. You drink because you are thirsty. The body, underneath that simple feeling, is solving a control problem with multiple sensors and delayed feedback. It must prevent dehydration without causing overhydration. It must protect blood pressure without overcorrecting pressure. It must conserve water without concentrating the blood too far. It must satisfy thirst before absorption is complete, but not so early that it miscalculates. It must keep the brain electrically stable while the body moves through heat, sleep, posture, meals, stress, and time. One glass enters that system. The system reads it. The brain changes because the control signals change. Not always dramatically, not always visibly, not always enough for you to notice. But the mechanism is there, and the emptiness of the stomach makes the lesson clean. A plain fluid enters an empty chamber. Sensors fire. The forecast updates. Thirst begins to fall. The gut confirms the input. Absorption begins. Osmolality moves toward correction. Vasopressin demand can shift. Kidney valves eventually change. Blood volume and vascular tone are managed. Attention may lose one background alarm. The brain does not drink the water. The body drinks it, and the brain reads the body. That is the quiet reversal. When you drink water before eating, you are not pouring fuel into your thoughts. You are changing the measurements that your thoughts are built on. The glass is simple. The machinery is not.